And also, I think here, I feel like I celebrate diversity. Not only you have here somebody like me talking funny, but I am the only one that probably can pronounce Carla's last name, okay? Then that's great. And then, on top of that, somebody's going to receive the Margalef Award, and my parents are from Spain. Then, you know, it's the whole shebang. Okay, now, it's my pleasure to introduce Carla Caceres. Carla Caceres actually is a product of the research experience for undergraduate programs. Her first paper as an undergraduate was published in Illinois, and she was telling me that an REU program was her door to become a successful scientist. Carla Caceres has been the recipient of the ASLO Linderman Award, and that award, as you know, is for a young person, a young scientist that writes an outstanding paper. Since 2001, she has been a faculty member in the University of Illinois Urbana, and right now she's actually the director of the Ecology, Evolution, and Conservation Biology program. In her research, actually, I think she has put zooplankton in the map out there. Her early work on the stages, the resting stages of zooplankton, kind of putting our minds how important it is to consider the true generation time of these individuals. And she has done many contributions in general population ecology and community ecology, using zooplankton communities as a model. And lately, and she will talk today about her work in disease ecology, again, in freshwater systems, and using zooplankton as her model. Then, without further ado, I would like to introduce Carla Caceres. And just a moment, I have to just retrieve my stool. And thank you very much for doing this for me.
All right. Is this mic on now? No, not yet. Is this mic on? Okay. Can everyone hear me all right? All right, great. Thank you, Maria, so much, both for that generous introduction and for the opportunity to come here and tell you today what this group of scientists is doing to study the ecology of infectious diseases. And what I'd like to do today is to first start by just giving you an introduction to the different types of diseases that we find in aquatic systems. Then move to a case study of disease in freshwater zooplankton that we've been studying, and talk about why we take this Forbesian approach that I've referenced in my title. And then finally end with a little speculation about what the future might bring. And what I hope you come away with from this talk is an appreciation for how taking an integrative limnological approach, one that considers both physics and biology, the biotic and the abiotic, the ecological and the evolutionary dynamics, can help us understand the distribution and abundance of disease. So if you start with the first question, why study disease? A short answer is disease is everywhere. And for those of you who attended the outstanding session yesterday on the ecology of infectious diseases, you got to hear firsthand the details of some of the specific systems. What I've done is just borrowed some pictures from the web to show you that we find diseases in freshwater, in marine habitats. We see phytoplankton infected with chytrids, infected corals. This freshwater snail has been castrated by its parasite. This Daphnia infected by a fungal host is what I'll talk to you in detail about today. The stickleback is infected with a parasite that will change its behavior in such a way so that the fish becomes more likely to be eaten by a bird, which the parasite needs to complete its life cycle. And finally, mosquitoes start their life cycle in aquatic habitats, and mosquitoes are important vectors for many diseases of humans. Here I'm showing the malarial parasite. So in addition to the concerns about disease and human health, we have prevalent diseases in a wide variety of different hosts that can influence both the ecology and the evolution of their host species. And increasingly, disease ecologists are asking this question: is a warmer world going to be a sicker world? As we increase temperature, are we going to both increase the distribution and the abundance of disease? And there's many reasons to expect that we might, and there's some good evidence for some systems as well that this might be the case. And I chose these data from a paper by Harvell, Wheel, and colleagues in Ecology, both for its clarity and for its local relevance. So what they're showing here is the prevalence of Caribbean yellow band disease, shown over here, from 1999 to 2007. And what we can see is not only does it clearly increase, but if you look at the average temperature and the mean prevalence, you see this correlation with warmer years having higher disease. So I'll return at the end of my talk to this question of, will a warmer world be a sicker world? So why this Forbesian approach? So I think about Stephen Forbes a lot. I teach in the building that he once occupied as chief of the Illinois Natural History Survey and professor at the University of Illinois. And given the age of the teaching equipment that I inherited, I honestly wonder if I don't have some of Forbes' original equipment. But what Forbes did in this paper that he originally presented in 1887 was to lay out this integrative framework for studying many different interactions in a lake. What Forbes did was not only link the individual to the community, to the ecosystem, talk about the biotic and the abiotic factors, but he even linked the ecological dynamics to evolution by natural selection. And it's this integrative approach that we and other disease ecologists are taking as we explore disease dynamics. What exactly am I talking about? So the study of disease is not new. People have been studying disease dynamics for a very long time. But one classical epidemiological approach to take is to start with a population of hosts that are susceptible to a particular parasite, and ask what it is about that host, that parasite, and the environment in which they occur that's going to move individuals of the host from being in the susceptible class to the infected class. So how does this disease move through this host population? And here you can ask, what is it about the host genotype or the parasite genotype? What is it about any immune response that the host might mount? What condition it's in? What about how the environment might modify that? But the take-home here is often this epidemiological perspective considers one host and one parasite. If we switch then to how a community ecologist might view the world, you might take that same species that we're thinking about as what we were thinking about as our focal host, and ask how it interacts with its predators and its resources and other competitors. Now, of course, host-parasite interactions are an interspecific interaction, so they fall into the realm of community ecology. But integrating host-parasite interactions and disease dynamics into this food web framework is a relatively recent endeavor. So what can we learn when we couple these different perspectives, and especially when we bring this evolutionary perspective to community ecology and acknowledge that different genotypes of the different players in this system respond differently to these interactions? So how does integrating all these particular factors help us understand disease dynamics? Well, there's increasing information from multiple systems that taking this food web approach, considering species other than your focal host and your focal parasite, can help understand disease transmission. What I've done here is I've taken this table from a recent review paper by Keesing et al. Just to point out, if we look at a range of hosts, we've got bacteriophages, we've got corals, we've got daphnia, we've got human diseases like malaria, we've got plant diseases. The take-home message is that diversity matters. So biodiversity loss can increase transmission. So when you have other species in the system, they modify this epidemiological response. The authors also point out that there are counterexamples where having other species may amplify disease. But the point is diversity matters, but what are the mechanisms by which diversity matters? And for that, I'll switch to our case study of diseases of zooplankton in freshwater lakes. So here, if we're thinking about diversity, we can think about different levels of diversity. We can think about diversity in the size and the shape of the lake, so diversity in habitat. We can think about the diversity in the community structure, And we can think about diversity in the host genotypes. Now, of course, these are all related, but I'll talk to you about how they are related. Now, zooplankton parasites occur everywhere from small rock pools to the very largest lakes. What we're talking about here are all Kellogg lakes in southern Michigan surrounding the all kettle lakes surrounding the Kellogg Biological Station. They range in depth from about 4 to 17 meters. But one important thing to tell you about our story here is how basin shape differs. So what I'm showing you here is a slide from Robin Smythe's dissertation. And what you can see is the bathymetric maps of both lakes with the depth contours in color. And the maps are overlain on their depth area curves. So we have some lakes that are more U-shaped. They have very steep sided walls, and much of the deep area and much of the lake is at the deepest point. Whereas we've got these other lakes that are more V-shaped. So they have much more gradually sloping sides and a much smaller deep area. Now we know that basin shape influences all kinds of things about a lake. It can influence its productivity, it can influence the community interactions that we see. And so what I'm going to do now is introduce you to the players in this food web and then talk about how basin shape interacts with the community players. So what we're dealing with here is a host, a focal host, Daphnia dentipora, that's common in Midwestern lakes, and its fungal parasite. So we can start, and we do start, with this optimal killer epidemiology. So there's many questions we can and do ask by just considering the host and the parasite. Daphnia become infected while they're foraging, and they eat these spores of the fungus. Those needle-shaped ascospores then puncture the gut wall and grow throughout the host. This makes infection very easy to diagnose because it changes the appearance of the Daphnia. It's obligate killer epidemiology because there is no recovery from this disease. Once a host is infected, it dies, and the host must die before it becomes infective to other individuals. So with this, we can ask questions about how different genotypes of the host respond to different genotypes of the parasite. How different temperatures modify that. How different foods. How the condition or the past exposure of the host might influence disease dynamics. But we know a lot about how Daphnia interact with other members of the plankton. So bluegill eat Daphnia. Bluegill are selective predators, and I'll show you in a little while, they selectively prey on these infected individuals. This sets up one of those mechanisms by which biodiversity can influence disease dynamics with this hypothesis called healthy herds predation. And I'll talk a little bit more about that in a minute. Fish aren't the only thing that eat Daphnia in the plankton. Daphnia also have to deal with predation by Chaabris. Chaabris are not selective predators on infected individuals, but they do influence disease dynamics. In fact, they may actually spread disease, and I'll come back to that as well. Daphnia eat phytoplankton, and there's work demonstrating that both the quality and the quantity of their resource influences how the disease spreads through the population. If we're talking about resources, then we also need to talk about competitors. Daphnia is not the only zooplankton out in the plankton. In all of these lakes, it co-occurs with another species, Daphnia pulicaria. Daphnia pulicaria feeds on these algal spores as well, but does not become infected itself. So what this can do, the presence of this other species, can again influence disease dynamics through this mechanism known as a dilution effect. So you have other potential hosts in the system that they themselves do not spread the disease as well. You can reduce the disease prevalence in any particular system. So when we put together even this oversimplified food web of the plankton, we see that there's many different interactions that could modify the disease dynamics in the system. So let me tell you a little bit about the spatial and temporal patterns of the disease that we observe. So as I said, we're studying these small lakes outside of Kellogg Biological Station, and here's an aerial picture of that with all the lakes in this lighter shade of purple are study lakes in the darker. We have circles around all the lakes where we have thermistor chains deployed during the ice-free season, and we have a Met station on Lawrence Lake. So we've been sampling 16 lakes since 2002, and what we found is that epidemics only occur in 8 lakes. So there's some lakes in which we never ever see this disease. So there's a strong spatial patterning to disease. There's also a temporal pattern, so more data here from Robin's dissertation. And what we find is that epidemics only occur in the fall. There's one more thing I have to tell you about this disease system. So this parasite has a problem. It has a problem in that its host is only seasonally available. So Daphnia and Typhra spend the winter in the sediment egg bank. They emerge sometime late spring, and they become abundant midsummer. We don't find the epidemic starting until periods of cooling later in the summer. And what I'm showing here are the infection dynamics in multiple different lakes in this one particular year. So the parasite doesn't swim. Its spores sink to the bottom of the lake. Its host is up in the water column. So what is it that could get those spores up into the host where they could get infected and start the disease dynamics, the epidemic for the season? Well, when we started this project, we'd been sampling these lakes for other reasons, and we knew that disease occurred some places and not others, and we knew that it was restricted to a particular time of year. So our original hypothesis was that basin shape would matter, and it would be the physics of the water movement that would start the epidemics going. So we hypothesized that we would see the disease occurring mostly in those V-shaped lakes. And where we came to that was, we thought that, well, the V-shaped lakes have more of their sediment exposed to the epilimnion. So perhaps resuspension could get those sediment spores up to where they needed to contact the host, and then we'd see disease spread. So, turns out we were completely wrong. Basin shape is correlated with disease dynamics, but in exactly the opposite way in which we predicted. So let me show you what's going on here. Here's the maximum prevalence of disease from 2003 to 2007. Each symbol represents a lake. The reason why there are these large error bars is because in some years, in some lakes, there's a really big epidemic, and then the following year there might be a very small epidemic. What we have on the x-axis is our metric of basin shape. And what we're using as our metric of basin shape is this metric we call Fee's probability. We took this from a paper in 1979 published by Everett Fee, where he asked what is the probability that a sedimenting particle is going to land on epilimnetic sediments versus falling through the thermocline in deeper water. And so when you have a low Fee's probability, there's a low probability it's going to land in the epilimnion. You have a very U-shaped lake out to a more V-shaped lake. So again, we hypothesized that we'd see disease in V-shaped lakes. We didn't. We see most of the disease happening in U-shaped lakes, and the largest epidemics that we see occur in those basins with the most steep, the steepest sided basins. So what differs between U versus V-shaped lakes? Well, there's a lot of biology and there's a lot of physics that differ between these lakes. So let me just tell you a little bit about what we know so far about how these lakes differ and what we think may be driving the spatial pattern of disease. So one hypothesis is just that the genotypes that you find in these V-shaped lakes are resistant to the parasite. But we can rule that out. We can create epidemics anywhere, anytime. We can take the host from the lake. If we add spores to that, we can infect individuals. So the hosts are susceptible in both of the systems, even though we only see disease in one. Another hypothesis might be that because we find more total phosphorus in these U-shaped basins, we would see increased disease. This is another hypothesis from the disease literature, that increased productivity leads to increased disease. But we don't see that in our system. I told you that resource quality matters, but when the epidemics are starting in all of our study lakes, the resource quality is sufficiently poor and doesn't really explain anything there. So what does differ? One physical mechanism that differs, and again, this is from Robin's dissertation with Sally McIntyre, is that in these U-shaped basins, we see the signature of gravity currents. So what these gravity currents can do is, as they come down those steep-sided basin, bring the sediments, the spores from the sediments out into the water column, where they're more likely to encounter those hosts and start the infection. We also think that there's biological reasons behind our pattern of spatial prevalence of disease, specifically predation. But as I told you, there's two different types of predation that's going on in the plankton. We have fish predation, which we think is lower in U-shaped basins. And we have predation by Chaabris, which we think is higher in U-shaped basins. Okay, so this healthy herds hypothesis is something that's not our hypothesis, it's in the general disease literature, and it's a fairly intuitive idea, especially for anyone who's ever watched one of those documentaries and you hear the narrator talking about the predator stalking the sick and the old and the injured and doing something for the good of the host population. And so what the healthy herds hypothesis suggests is that if predators selectively cull infected individuals, they can keep a disease either at low abundances or from ever getting started in the first place. And again, bluegill eat Daphnia, and bluegill selectively forage on things that they can see. And to remind you that when a Daphnia is infected, these spores build up in a heart throughout her entire body, and so even to us, it really changes how visible the Daphnia are. As part of her thesis, Megan Duffy caught a lot of fish. She looked at the abundance of the infected individuals in the guts of the fish versus the number of infected individuals in the water column, and calculated Chesson's alpha, or how selective are these fish on these infected individuals. And the bottom line is they're incredibly selective. There are times when the epidemic would only be in a few individuals in the water column, so just getting going, yet you would find these fish that were just completely packed with the infected individuals. So how this relates to our spatial patterns of disease is we know as limnologists that when you increase predation by fish, you often see a correlated decrease in the size of the body size of the zooplankton. So if we look at the mean adult body size of our hosts in our different lakes represented by each point, what we see is a significant decline in body size of the hosts as we move from more U-shaped lakes to more V-shaped lakes. So from this we infer that the vertebrate predation intensity is higher in these V-shaped lakes and maybe keeping disease at bay there. Chaabris is different. Chaabris is not selective on these infected individuals. And if we look at Chaabris abundance against our lake shape, again we see that we have more Chaabris in certain types of lakes, these U-shaped lakes. Now, based on what I just said about vertebrate predation being higher in V-shaped lakes, this makes sense because Chaabris can be eaten by fish as well. When we look at Chaabris density and the maximum prevalence of disease, again the large error bars because the maximum prevalence every year varies, we see a positive correlation. So more Chaabris is more disease. Is this a correlation or is this causation? To ask that question, we ran a series of laboratory experiments. We started first just in a flask, very simple experiment where we had two host populations, infected both of them, put Chaabris in some and not in the others, and at least in a flask, when you put Chaabris into a flask, you see higher prevalence of disease, both if you just consider the adults and if you consider the adults and the juveniles. So at least at a small scale, Chaabris can spread disease. But by what mechanism? What we hypothesized goes back to this problem that the parasite has, in that it needs to be up in the water column where it's going to encounter its hosts. So if you have the infected individual up in the water column, and it just dies of the disease, somehow it managed to escape being eaten by the fish, it sinks rapidly where the spores are then deposited in the deep water sediments, where they may never infect another individual. In contrast, the way Cayabrus feed is that they ingest the prey item, take what they need, and then they regurgitate. Here's my poor little Daphnia regurgitated. We also hypothesized that they could regurgitate spores, and by doing so, keep these spores up in the water column where the other individuals might contact them. So to address this question, what we did was we set up another laboratory experiment in which we first took just infected Daphnia and let them die from the disease. They were immediately removed from the beaker to simulate sinking out of the epilimnion. From that, we counted the number of spores that were released in the short time that they remained in the beaker. And what you can see is there's very few. So this is the worst case scenario for the parasite because all those spores are now on the bottom of the lake. If you let Cayabrus eat the Daphnia, infected Daphnia, you increase the number of spores that are released. So in this treatment, we remove the regurgitant. In this case, we ground it up to see how many of the spores were still remaining in that carapace. So we continue to go up. So some spores are maintained in that regurgitant. And finally, we just let some individuals die of the disease to grind them up to see what fraction of the spores were being released. And so the Cayabrus, either by regurgitation or defecation, releases some spores into the water. Not 100%, but significantly more than if the Daphnia would just die. Importantly, these spores remain infective. So we then took these spores and transferred them to new hosts and found that they still could infect the Daphnia. So habitat matters. We've got this U versus V-shaped pattern. Predators matter. But predators matter in a different way, depending on their identity. What about host genotype? What I'm showing you here is some data from my master's student, Chris Bertram, and I've plotted the reaction norms for 2 different genotypes of the hosts in a variety of pheromone treatments. There's a couple of things I want you to take away from this slide. The first is that some genotypes are more susceptible than others. So the blue line is always higher than the red line. This one gets infected a lot more easily than this one. The second is that I told you that predator identity matters, and there's both those direct effects of how they feed, but there's these indirect effects as well. So these hosts are just grown in the water with the predator. They're not having any direct interaction with the predator, but they're still responding to the predator. This is not surprising. We know that Daphnia genotypes respond to pheromone treatments. But we can have, and I'll show you in a minute with more data, different responses to each predator. So genotype matters. The predator identity matters both through direct and indirect effects. And then when we now each one of these lines is a different genotype onto the slide, what you can see in this mess is basically we have a lot of genetic variation, both in how the parasite is transmitted through that particular genotype and how the different genotypes respond to the predator. So this is just a little bit of data to show you how we're considering these potential for evolutionary dynamics also interacting with the ecological mechanisms that I talked about. So hopefully I've convinced you that different mechanisms are working together in determining the distribution and abundance of disease. So habitat matters, community matters, and genotype matters. As I said, I was going to return to the question of what will the future bring. And as we heard yesterday in the Ecology of Infectious Diseases session, that there are several systems for which it seems very clear that a warmer world will be a sicker world. For ours and for others, the jury's still out. And for us, it has to deal with how climate change is going to influence the outcome of all these different interacting mechanisms. So climate change isn't going to influence the shape of the basin, but it is going to influence stratification patterns. We see in our data signatures of correlations between the strength of stratification and the timing of the onset of the disease, as well as its magnitude in a particular year. Warming is predicted to have both direct and indirect effects on community interactions. And so as we change what the community structure looks like, might we change how disease moves through the system? We also need to consider the fact that each player in this food web has its own thermal ecology. And one thing Spencer Hall pointed out in a theoretical paper he published in 2006 was if you consider just the fish predator, the Daphnia host, and the fungus, and if you think about how their physiological rates scale with temperature, in cases such as ours, where those vital rates of the predator scale more steeply with temperature than does that of the host, what you see is as you warm, it becomes easier for the predator to control the disease. So in that case, warmer doesn't mean sicker, warmer means a reduction of disease. But then how that fits in with these other interactions, as well as what we might expect to see in terms of changing host genotypes as we change both the thermal regime and the community interactions, is going to be what's going to answer the question of, for certain systems, will warmer be sicker? And as we move forward with this, we not only continue to integrate the biology and the physics, the ecology and the evolution, but also with an explicit focus on how warming might influence those different interactions. So I just want to end then with once again acknowledging my collaborators. This really has been and continues to be a group effort, and thank you very much.